today. So today I'm going to talk about some of the work we've done at Cornell since I started my group. And the title is Genomes as Graphs. And so primarily I'm going to talk about things related to ancestry recombination graph and a graph that's similar to ARG, that is a graph that we created in my group. So I'm going to start with showing you this picture from the UK Biobank that in 2023, they released this whole genome sequencing data of 500,000 individuals' genomes. And on their website, they say like it has generated 27.5 petabytes of data. So we have a lot of data now, at least in human. And in other species, it's also a growing amount of data. So what are the data we care about as population genetics and statistical genetics? So the data we care about are actually the genetic polymorphism data. And here I'm showing 2 DNA sequences. Apologies. So 2 DNA sequences, and you can see in this alignment, here there's a GA polymorphism, and that's where these 2 sequences differ. And at population level, that's the information that could cause selection, that could cause phenotypic differences among individuals. And since we study diploid genomes, we can have homozygous A and heterozygous and homozygous G. And if we use numbers, integers to indicate it, we could use 0 and 1, and 0 to indicate the reference allele or ancestral allele, and 1 to indicate the derived allele or the mutation. We could also use the counts to indicate the information. So we could have 0 copy of A allele, 0 copy of alternative allele, 1 copy or 2 copies. So these are the ways we can indicate the data and we can utilize it. So a lot of you use isogenic lines here, in which case you get this phase genome automatically. But in human data, we actually start with getting the data that is unphased genotype. So in the VCF format, there's a backslash here indicating it's unphased. But statistical phasing algorithm could now use a lot of samples to phase these samples. So at the end of the phasing, we will get one of these possible phase of the genome, and with this vertical line separating the 2 alleles, indicating that the left-hand side belongs to one haplotype and the right-hand side belongs to another haplotype. And this kind of statistical phasing method right now is very scalable, so you could actually statistically phase 500,000 genomes, and that's no problem. So phasing the Biobank-Scale whole genome sequences can be done, and it would still cost a lot of money, like 10,000 pounds or so to do this, but it's a one-time cost. So in the UK Biobank, we have this data readily available. So yeah, so in terms of encoding this kind of polymorphism data at population scale, the most popular format is VCF. And VCF is a very flexible file format. So what people use for downstream analysis is this PVCF. That is the VCF format for population level data. And we could have this unphased VCF format or phased VCF format. And starting with this data, when we do the analysis, we usually convert it to this 0, 1, 2 encoding. So it works as if it's a matrix, and then we can do downstream genome-wide association study or some population genetic analysis. This particular format is highly intuitive, and we can utilize highly optimized algebra library for computation. However, as the data sets continue to increase, there's a very high cost for input and output, and it also it takes a lot of space, and it doesn't utilize the correlation among the SNPs or samples. So it actually takes the entire space and grows with the number of samples and grows with the number of variants in the data. So so this is our rough estimate of how much data we have, and so there are a few large biobank cohorts that's available for researchers to use. And collectively, there are more than 1 million whole genome sequencing data for humans. And if we use this VCF format, then collectively it would take approximately 1.7 petabytes of the space to store it. And what people do these days is to use this compression algorithm that's a common compression algorithm called gzip to compress the data. And then after that, you get this vcf.gz, and which is quite a bit smaller, but still it would take 11 terabytes of space to do this. But however, this only partially solved the problem of how to save this data on hard drive. But when you analyze the data, you still need to decompress the data, and then this data becomes larger again when you analyze it. And then there's still this problem: how do we analyze such a large data set? So what this biobank gave the solution is that let's do cloud computing. So right now, this access to this data are restricted to the biobank cloud computing platform, and as a result, it would cost more money for researchers to do any analysis, and for people who does explorative research, it's very hard to start exploring this kind of data. So as someone who comes from a background of algorithm genetics, I used to deal with a smaller scale data set that's ready for analysis. So what's really desirable for me is like, oh, could we still analyze this large data set on a small computer resource like a laptop or a local server? Is that even possible? So there's this concept in computer science called succinct data structure. So it's talking about this hypothetical data structure that use the amount of space that's close to the information theoretic lower bound, but different from a regular compression method or a compressed representation. It would still allow people to do efficient query operations. And the reason for this is that this data structure is supposed to be related to the data generating process. And because it's related to the data generating process, it can compress the data, will allow data query operations. And data query operations is what we need if we need to do any sort of computation or analysis. So if such a data structure exists for genetic data, then it could improve the computational scalability and allow us to save the data very compactly. And it could potentially also allow biological interpretation because that's supposed to relate it to how the data is generated. So that's probably something we would like to see, and that's the direction we were thinking about. So to start, I'd like to talk about something that everyone's familiar with. So if we have a DNA sequence alignment, like what's illustrated here, we could build a phylogenetic tree. And if we have this phylogenetic tree, we could also ask where are the differences, like where could we place them on the branches on this phylogenetic tree. So these differences in the DNA sequence alignment could be put on these branches, and that would indicate whether that's a unique mutation to human or a common mutation between human and chimp, for example. This may be gorilla rather than chimp, I'm not sure. So we could efficiently keep this alignment information on top of phylogenetic tree, and without keeping this redundant information. We only need the ancestral sequence, right? So we can see that a phylogenetic tree structure could be used to store this data efficiently by just recording the mutations and the tree structure. So in population genetics, we have this concept called ancestral recombination graph, which is a collection of trees along the genome. And this data is based on individuals within the same population. So within the same population is no longer a single phylogenetic tree structure because within population, there's mating and there's exchange of genetic material. So it's not a single tree, but rather a collection of trees. So from one position in the genome to the next, the tree structure could change because the genealogy ancestor could change. So to give you an example, so between my cousin and I, we would share two grandparents, and that's going to be our last common ancestor position in some parts of our genome. But we also have grandparents that we do not share. And in those parts of our genomes, our relatedness is actually pretty similar to two random samples in the population. So that reflect on ARC, it would be like two leaf nodes that's closer to each other in one part of the genome, but then they are further away to each other in another part of the genome. So along genome, this tree structure will change. And but if we had this kind of graph, we could also imagine that we could put mutations on top of this graph, and then collectively that would save the DNA sequence information. So potentially ancestry recombination graph is a data structure that could efficiently save the genomic information. However, it currently couldn't provide a solution for biobank-scale dataset for two reasons. One is that that ARG inference is not scalable enough for biobank-scale whole genome dataset. So you could infer ARGs for a fraction of the biobank scale samples, or you could infer ARGs for a fraction of the size from the whole genomes. But currently, we don't have any method that could allow us to do this inference for whole genome sequence data and for all the samples. And another reason is not all ARGs would encode the mutation information. So some ARG inference methods do not encode this information because they don't care about this information. Some ARG inference methods do not care about encoding the same information exactly. So they only approximately put the mutations on the node because not necessarily the mutation could be put there. So that's another reason. And the third reason we think is probably not the format we're looking for is that not all computations can be done efficiently on ancestral recombination graph, especially if we care about statistical genetic analysis, then we want efficient linear algebra operations on top of the graph. So what we came up with is this genotype representation graph. And to briefly introduce this, we also have nodes on this graph. And these leaf nodes would represent haploid genomes or haplotypes, haploid individuals. So if we have a diploid individual, it would become two leaf nodes, similar to ARG. And we would have these common nodes above two leaf nodes, and these are internal nodes. And we would also have root nodes above these nodes. And so that in some sense is very similar to ARG. So we specify that root nodes must contain at least one mutation. So when we say a node contains a mutation, it means that this mutation so we have some mutation put on this node, it means that this mutation would pass down to all the descendants of the node. And if a node, let's say this node is not a descendant of node 7, then this node wouldn't have the mutation. So this is the rule we developed this graph. So there must be one and only one path from the mutation node containing a particular mutation to a sample node if and only if the genotype for that sample node contains that mutation. So I'm going to show you what I mean a little bit later. So what do we call mutation? So mutation could be derived allele, or it could be the minor or alternative allele. And the null allele, it would be then the reference allele or the ancestral allele. So what we encode as a mutation, we do the coordinate and allele information to encode this mutation. So if we have a biallelic polymorphism, then it's encoded as one mutation because the reference allele doesn't need to be encoded. And then if we have a k-allelic polymorphism, then it will be encoded as k-1 mutation. And if there's missing data at a particular SNP, then it will also be encoded as one mutation. So that way, we have the same information as a VCF, as a hard call VCF file. So how do we query a mutation if we have a graph like this? So on the left-hand side, we have a table of haplotypes. So each row is a haplotype, and each column represents the information about this mutation. So we have 4 haplotypes here on this 2 red box area. We see 1011 information that they're identical. So this means that these 2 SNPs or mutations, they're in complete LD with each other. And on our graph, these 2 mutations are put onto node 7. And we can see that node 7 have descendants node 0, 2, and 3. And therefore, it means that node 0 and 2 and 3 would have this one allele information, and 0 information is implied by default. Therefore, we have the same information about mutation 5 and 10 as the left-hand side. And similarly, this graph has exactly the same information as the left-hand side. We can use this graph to represent the same information. So that's how we query a mutation from this graph. It's simply a downward traversal from the mutation node to the sample node. So how do we query a haplotype? So as I mentioned, these leaf nodes represent a haploid, a genome, or a haplotype. So in this case, if we want to know what is the haplotype at leaf node 0, we simply need to collect all the mutations that's mapped to this particular node. They are singleton variants. They are singleton variants because they only occur on this leaf node. And then we traverse upward to node 6, we traverse upward to node 7, and we collect all the variants on these two nodes. And the rest of the information are implied. They are the same as the reference. So similarly, if we want to know what is the haplotype at node 2, then we traverse upward, collect all these mutations on the way, and then we reconstruct the information on this haplotype. So you can see that we can query the information which samples have which mutation, and we can also query which mutation get passed on to which sample. So that's simply a downward traversal versus an upward traversal in this graph. So since traversal can be upward or downward, that's pretty much the same thing when we have a graph. The GRG graph represents both the haploid genotype matrix G and its transpose, G transpose. Because mathematically, or if you code it, if you program it, you can actually only easily access either the row vector or the column vector through programming most efficiently. But in GRG, these two are symmetric. So I talked about the property of GRG a little bit. Now I'm going to briefly introduce how we construct it, because the goal of this project is to be able to construct it for biobank-scale dataset, and then to see if we can do some analysis much faster than before. So we start with this phased genotype matrix, and we want to construct this graph. So briefly speaking, we first split the genomes into different segments, and each part is not too long. So, and then within each split, we would build a graph shape based on sequence similarity. So we get this graph shape built based on the similarity of the sequence, which means that a more similar sequence would tend to have a node above them that's closer to them, and a more different sequence would be further away on this graph. And after that, we would try to put mutations onto the node. So each node we already have would correspond to a set or some sorts of information. It would correspond to which sample node have the information passed down from this node already, right? So based on that information, we will try to match with the information of each SNP and then make a one-to-one match. If there's a one-to-one match, then a mutation will be put onto a node. And in some cases, there may not be a one-to-one match, which means we need to create a new node to put this mutation there. And in that case, we would try to minimize the number of edges we add to this graph and try to utilize as many previous nodes as possible when we construct this new node. And after that, we could merge the graphs. This is not entirely necessary, but if we merge the graph, we get a single fully connected graph. And then we have a step that is simplified to remove some useless nodes that would only make the space larger. And then we encode it in this compressed sparse row format as in the disk. So since the publication of our first paper, we made, Judy has the programmer in my group made some further improvement. So the result I'm going to show you are actually new results, both the published version and this version 2, which is even smaller than our version 1 by 50%. And the property of this version 2 is even better because we now have the same graph format in hard drive and in memory, which means when we load the data from disk to RAM, we don't need to do any actual construction of the graph. We can analyze it as is. So I'm going to show you some results. So first, I want to show you, how do we do in compressing the data in terms of the file size. So the very left panel is based on simulated data. So x-axis is the number of samples. It varies from 10 to 1 million samples. And the y-axis is the size of the data. So let's look at this lowest line. That is the GRG version 2. And the highest line is this VCF. And then in between them, there's this compressed VCF, vcf.gzip. And then we also have this Plink format that's quite commonly used in structural genetics. So we can see that when sample size is large, the GRG version 2 is really much smaller than these other alternatives. And then this middle panel, what I'm showing here is the result from application to the UK Biobank dataset, and this lowest line is the GRG version 2, and each dot here is a single chromosome. So we use 200,000 phased genomes from the UK Biobank, and each dot is a single autosome here. And this third panel is really just a zoomed in version of it. So if you want to see numbers, you can see like for chromosome 2 is the largest in terms of file size because it has a lot of variants. And for that particular chromosome, it takes 5.3 gigabytes space for the version 2 GRG. And if we use the compressed VCF, it will take approximately 190 gigabytes of space. And for the whole genome, for all the 22 autosomes, to be more specific, it would take 66.8 gigabytes for GRG version 2. So compared to the compressed VCF, it's really much smaller. And it could now really fit into a laptop space. And per chromosome, it can easily fit into any laptop's RAM space. So that's the size, right? But how about computation? So because in GWAS, what people care about is this computation based on diploid genome, because we have phenotype measured based on diploid genome. But what we represent as a graph is this haploid genome's information. It's one-to-one compared to the haploid genome's information, but to convert the result to the diploid version, we need to do a little bit of additional work. So what we could do with this graph is the dot product between the haploid genome matrix that we call G with a vector or its transpose multiplied with a vector. So the haploid genome genotype matrix has the dimension of 2n by m, where n is the number of samples, m is the number of variants. And the original diploid genome is this n by m genotype matrix, and n is the number of diploid genomes. So each row in this original X matrix is expanded into 2 rows in this haploid genotype matrix. And that haploid genotype matrix is what GRG could represent and what our computation could do. So which means that when we have this dot product computed based on GRG, we need to find mathematically equivalent result that gets us back to the result based on this original genotype matrix X. And that way, we can get the same result as before. So to give you an example, so let's say we have a diploid genome X, and we have a haploid genome G. If we simply want to compute what is the total number of counts of the minor allele for each SNP, it's simply this matrix multiplied with a vector of 1, because it's a summation of all the elements in each row. So it's this matrix multiplied with this vector. And in the diploid version, we would multiply with this vector of size 1 with size n, and in the haploid version, we would do this with 1 with the size 2n. And we know that this G transpose multiplied with a vector of 1 is something we can compute on this graph as a dot product. So it's really just adding things up as we traverse the graph, as we move upward. So when we move upward to the graph, we add things up. That way we reuse the calculation we already did for the lower node, because this graph has this hierarchy. And the number of calculations is the same as the number of edges in this graph. If we do it in the matrix format, we need to loop through each row and each entry in each row. So that's how we do this calculation. So here, what I show here is the time comparison. So again, this x-axis is the number of samples, y-axis is the time. So this light blue color is how this allele frequency calculation works with GRG. And we can compare to this other format, including TS-KIT, that is based on ancestry recombination graph, and including Plink. And we can see this dot product calculation for allele frequency, or just allele frequency calculation, is the fastest. And it's about 10 to 100 times faster than these alternatives. So this second panel is application to 2 different chromosomes for the UK biobank, and we see a similar trend. Okay, but dot product calculation for allele frequency is just very simple. So we want to see if we can do a little bit more. So another somewhat simple calculation is how do we simulate phenotype, right? So to simulate phenotype, we have this original diploid genotype matrix X multiplied with the causal effect vector beta, and then plus some environmental noise. So that way we get this phenotype simulated that is represented as Y. So in our graph, we can compute this matrix G multiplied with this beta. And beta are the effect size of the mutation. So some of these mutations on this node would have some effect. So how do we compute this G multiplied with beta? That is a downward graph traversal based on this topological order. So if we move from node 8 to node 7, we would first need to assign a value to node 8. That is whether this mutation on node 8 has an effect. So if it does have an effect, we assign this effect to the effect of node 8. And then when we move to node 7 would collect the effect from the node above it, that's node 8. It would also collect the effect from M2 mutation, whatever causal effect this mutation has, it will add to that value. Then that way we calculate the effect of node 7. And then as we move downward to node 9 would have its own effect from mutation 1. And then it would also collect the aggregated effect from node 7. So when we move to node 9, we would add the beta 1 to mu7, that is the node effect from node 7 with the node effect of node 9. So then this effect is passed down to node 4 and node 5. So you can see as we move downward, we have some calculations that don't need to be computed again, because when we do this on the graph, it will reuse some of these calculations. And after we have this G multiplied with beta, we have each of these genetic value computed for each haploid genome. So so then 2 of the haploid genome belongs to a diploid genome, and that means we need to add up the genetic value from the 2 haploid genome and then combine them into the genetic value of the diploid genome. And after that, we add the environmental noise to this genetic value. We simulate the phenotype. So Again, we compare with TS trait, that is an ancestral recombination graph-based simulator that was just published last year, and is supposed to be the fastest phenotype simulator. So compared to TS trait, we can see when sample size is large or when the number of causal mutation is large, we are about a few hundred times faster than TS trait. And we also are interested in doing genome-wide association and more complicated calculation. But the principle of this calculation is simple because what we derive is how to do this haploid genotype matrix multiplied with a vector, this matrix dot product. And we just need to convert this original computation with respect to the diploid genotype matrix to this thing that we can compute with this haploid genotype matrix. So to do genome-wide association, we start with this single linear regression coefficient and all these statistics associated with it. And it becomes this dot product calculation, and that's something we can compute. So what we implement is the same calculation as the Plink association flag. And we confirm that we get identical result to this Plink software. So again, on the left-hand side is the simulation result, and right-hand side is the application to the UK Biobank based on 2 chromosomes. So we can see that this yellow line is the result from using Plink with Plink format, and this light blue line is the GRG result. So we are magnitudes faster than this Plink software in doing genome-wide association. And at the time of our paper, we haven't figured out how to do GWAS with covariates, but we recently figured that out. So we will have this full version of GWAS pretty soon, hopefully by the end of the summer. And we keep enriching our software as we add new features. So that is GWAS. So GWAS is still considered a relatively doable thing, even with large data set. You just need, to put each phenotype to each node, and then you take up the resource. So it does cost a lot of money when you do biobank-scale thing, but it's not considered something that you couldn't do at this scale for whole genome. So we're also interested in something more expensive. So the question we try to deal with is this fine mapping problem. So what people usually consider a genotype-phenotype map is that along the genome, most of the SNPs do not have a phenotypic effect, but there are a few causal variants that would have a causal effect. So that's what people think is the ground truth model. But in GWAS, because of the linkage disequilibrium effect, what will be seen is that if we do this association, then there will be peaks and a lot of signals cluster in together in this region. So it's very hard to know how many causals are there and what are the causal effects. And fine mapping try to disentangle this messy signal and try to get more clean signal that there are how many signals there are and how many could be explained by a few variants. So one of these recent popular and quite scalable fine mapping method is this sum of single effects SUSY model from Matthew Stephens groups. So in short, this method would model the genetic vector as the sum of multiple single effect regressions. And in each of these single effect regression steps, it will evoke estimating the association effect and the posterior inclusion probability for each variant. And then it will iteratively update these signals and then until convergence to try to get the number of signals and then estimate a credible set, that is, what are the variants that potentially explain the signal. So each of these single effect regression step is very similar to a regular association step. And that's something we know we can do very fast with this GRG GWAS approach. So we want to try, okay, could we just do the same thing as this SUSY model and then see if we're much faster. So we pretty much do the same thing line by line, but with our library compared to this original R package. So to implement this. And here's the result. So on the x-axis, we have sample size, and y-axis is a run of this fine mapping approach. And this is based on a particular simulated architecture, but we do expect that we will be generally much faster. And we can see that when sample size is large, we're really pretty fast. So in this particular setup, it would take more than 6 hours to run SUSY, and then it takes only 30 seconds to do the same thing, get almost exactly the same result with our approach. The minor difference of not being exactly the same result is because we would collapse exact LD into one regression, rather than if you have a genotype matrix, if you have complete LD, you would do 2 single regression to these 2 signal. And then as a result, the order of the things could slightly differ, but pretty much you get the exactly same result with this new approach, and it's much faster. And we're interested in exploring whether we can improve fine mapping as well. So this is simply a speed comparison. So I hope I provided some evidence that we can do things much faster, but that's all things people can already do with other software, right? But I think GRG does hold some potential for doing things that we previously couldn't do because it's just way more expensive. So there's this question: what is a GRG really? So in computer science, actually, there's this data structure called multi-tree that has been proposed and used, and GRG belongs to that particular type of data structure. So it's this, it is a directed acyclic graph, but it's more special than that because this multi-tree data structure has this feature that below each node, in the downward direction, you have a tree structure. And let's see here. So below this node, in the downward direction, you have this structure that's a tree. And above each node, in the upward direction, so if we go upward from here, we also have a tree structure. So every node in this multi-tree structure has this feature that below each node, you have a tree, and above each node has a tree. So the base of this multi-tree structure is a tree. So the question is, why is GRG like that, and why could we compress the data this way? So below each node is a tree, right? So below each GRG node, we have a mutation and how it gets passed on to the descendants. So this is very intuitive because coming from ancestral recombination graph, we start with a sequence of trees, and then there are mutations on the branches. So below each mutation, we can see a subtree. So that kind of tree structure could come from a coalescent tree and how a mutation occur. So that's below each node. What about above each node? So if we think about haplotype inheritance within a pedigree, we can start with an ancestral generation. This is a diploid, and each haplotype is colored in a different color. And then there can be recombination between these 2 haplotypes, which generate a haplotype that gets passed down to the next generation's individual. So we do this for all of these individuals, and then 2 haplotypes would merge together to form a diploid individual, and then there will be another round of recombination between them to generate a new haplotype. So at the end of this, we get a focal haplotype that's a mosaic pattern from its ancestral haplotype. If we try to trace these color blocks back to the ancestor in this earlier generation, we can see that not our ancestor would pass down genetic material to this focal haplotype, but more than that, we see a tree structure start to emerge. And in this tree structure, there's this merge of lineages passing down, and that is recombination, combining different genetic material that was originally in different ancestor, but that gets merged together into a recombined component that could potentially pass down as a single unit until further recombination break it. So that recombination And that kind of structure going backward in time is a recombination splitting genetic material up because they come from different ancestors. That explains the tree structure above the node. And that is what we have here as this tree structure above the node in the GRG. So in some sense, we think of this GRG structure, it could come from a reduced version of ARC. So we don't have all the information in ARCs like coalescence time and this other information. And in some sense, we're also, we haven't really tried to infer it, but there could be a GRG that is more related to the data generating process. So in our construction, it is a greedy algorithm that we just want to scale it up to biobank scale. So it's nowhere near that true structure, but potentially related to this genealogical process, there should be a reduced graph from ARC that would be like somewhat closer to the true history of how haplotypes are created by mutation, and then how they pass down, and how recombination form new haplotype. So this is a reduced graph from the ARC with somewhat less information, but it keeps the information that actually passed down to the present day samples. And in the future, we're also interested in seeing if we can actually build a graph that's more related to the haplotype history rather than just to compress the data. So we talked about GRG can compress the data and we can do things much faster now. So potentially that could allow analysis being done on laptops or local servers. But so in some sense, I sort of think of it as it is a data structure that can democratize the data computation, because computation now is so expensive. So not everyone could afford to do biobank scale analysis. And there's this other question that I think could further democratize the data access. That is, what if everyone could access biobank-like data? So of course, we cannot really release human genomes with this consent to the public, but maybe we could benefit from having data that look just like real for not only educational purpose, but maybe also for research purpose. So about this, we have to think about how to simulate data that's more real than what we could simulate with a population genetic simulator or with a machine learning model. So with population genetic simulator, what happens is, even though we can simulate genomes based on some parameters, they don't have the same distribution as the real data. You don't have the same mutation, you don't have the same amount of LD, and then in some sense, is not mimicking real data. It's trying to regenerate some data, mimicking some process, even though it's very helpful. It's not real data-like. And machine learning method could also generate new data, but there's the question of how you can simulate new variant with a machine learning model, because you have this training data, and then when you generate more data, it's based on the mutations already exist in the training data. Whereas we know that if we only do that, that's not going to mimic real data. So the solution, or like what we propose as a potential alternative solution, is that maybe we can utilize the ancestral recombination graph and then simulate based on an ARC. So a student of mine, Jinmin, so he derived a conditional coalescence-based recombination model that allow us to sample new haplotypes conditional on an input ARC. So on this left-hand side, we have an input ancestral recombination graph, and then which could either be simulated or it could be inferred from real data. And then what he did is to conditional on this input ARC, and then to simulate some samples, some new samples, and then add these new samples, thread these new samples to this input ARC. And then these new samples would inherit mutations and recombination and coalescence from the input ARC, but they would also have their own new structure. And then we can remove this input ARC and the input data, and the resimulated data would try to follow the same process, inherit the same pattern of mutation and LD pattern, but there would also be realistic new mutation being simulated based on the branch length. So here's a proof of principle test. So we have the input data that is just 100 genomes from 3 simulated populations, and then we can resimulate a lot more data based on this approach. So somewhat different from machine learning approach is that we don't need a lot of data to simulate a lot more data, because in this process we have a generative model for new mutations and recombination, so we can generate way more data than what we originally have. And so we can also test the allele frequency distribution based on all the SNPs and the LD pattern based on all the SNPs, and we expect them to be really close to each other. And we can also test based on coalescence time, because at the end of the simulation, we do not just get the simulated DNA sequences, we also get a simulated genealogy. And in some sense, if we really want to remove the existing mutation, we can also do that. We just overlay based on mutation rate on this new simulated genealogy if we're concerned about the privacy of these common variants. Okay, so in summary, so I talked about GRG could work quite efficiently for compressing the data. And for each chromosome for biobank-scale dataset, it can now fit into the RAM of a regular laptop. And GRG matrix multiplication is extremely fast. And we have a Python API that also make it quite easy to use. And based on this GRG matrix multiplication, we built GRG phenotype simulator and GRG SUSY, which are also much faster than the alternatives. And at the end, we talked about that maybe we can simulate new samples based on an input arc. So with that, I'd like to thank all the people who work on this project. So Drew Diaz is a programmer in my group. He's the main developer of the GRG library. And Drew and Ziqing work on co-led the first paper on this graph structure. Aditya Syme was an undergraduate student who just recently graduated, and he built the GRG phenotype simulator. And Bella is an undergraduate student, and she worked together with Ziqing to build the GRG SUSY. And Jinmin is a first-year graduate student who worked on the ARC-based simulation. And thank you, and I'll take questions.